Seja bem-vinda ou bem-vindo a mais uma sessão do Congresso Acadêmico Unifesp 2020.
So good afternoon, everyone. Thank you so much for being here. We are going to start our session on neuroscience with the theme Neurotechnologies for clinical rehabilitation in sensory motor disorders. Well, the session is going to be in English in respect to our guests. And I'm sorry, there was some problem here. I would like to start by thanking the whole support given by Thais Toledo, Daniel Leal, my PhD students, who are helping me to coordinate everything here in the backstage. I also would like to thank Professora Gabriela Brelas to invite me to prepare the session, and of course to Unifesp. So I'm very pleased to announce, without overstatement, two of the greatest researchers in the field of neuroengineering and sensory motor disorder studies, Dr. Soleiman Shokur and Dr. Silvestro Missera. So thank you very much for being here to accept this invitation. I'm very glad that we could make it real. So just to explain to the public how it's going to work. So first we have Dr. Missera presentations, and then we follow to Dr. Soleiman. Both of them we're going to have around 30 or 40 minutes, and then we open for questions for everyone who is interested to question something for both of them. So all questions has to be made by the chat, or maybe also through Facebook. Danielle is there compiling everything and organizing, and you can type the questions in Portuguese as well, and then we can try to translate it for them. Okay. Although Soleiman can understand very well Portuguese, so you can also abuse him. So let me, for those that don't know yet, let me introduce the first speaker, Dr. Missera, to go straight for his presentation. Dr. Missera received his degree in electrical engineering from University of Pisa, and his PhD degree in biomedical engineering from Escola Superior Santana. From 2000 to 2009, he has been an assistant professor of biorobotics at Escola Superior Santana, where he's now the head of neuroengineering group. In 2007, he was visiting scientist at Massachusetts Institute of Technology in USA. And from 2008 to 2011, he was the head of the neuroprosthesis control group and an adjunct professor at Institute for Automation in Swiss Federal Institute of Technology in Zurich. In 2009, he received the Early Career Achievement Award of Engineering in Medicine and Biology Society. And since 2011, he's associated professor and head of the Translational Neuroengineering Laboratory at EPFL, École Polytechnique Fédérale de Lausanne in Switzerland. Dr. Missera's interests include the development of neuroprosthesis based on the use of implantable neural interface with the central and peripheral nervous system to restore sensory and motor function in people with disabilities. Well, the list of achievements and prizes is endless, and I think it would be better to hear directly from here by his presentation. His presentation is called I for Life, Intraneural Stimulation to Restore Sensory, Motor, and Autonomic Functions and Improve Quality of Life. So thank you very much, Dr. Missera, and well, the word is with you right now. You can start when you feel good.
Muy Obrigado. Now falo Portuguese, so I'm going to speak in English. I apologize. I just used only few words that I know, that Google Translator told me to say. So I hope you can hear me now. I'm going to start very briefly with kind of a very short introduction about what we call neurobionics or neuroprosthetics, which is the idea of restoring connection between parts of the nervous system, between the nervous system and the external world. And in some way, what people like me or Soleman do, we try to be the bridge between basic science, in particular neuroscience, and clinical trials. So to speed up the transfer of basic know-how learned in neuroscience into real clinical applications. In particular, the message we want to convey you today is that with neuroprosthetics or neurobionics, the number of things you can do is almost unlimited. So if you're able to play, as one of the pioneers in the field, Tom Mortimer, was used to say, or he told me when a young PhD student many years ago, if you can play with the nervous system, you have unlimited possibilities. So today I want to give you a couple of examples, and Solomon will give you even more in this direction. But at the same time, if there are young students willing to know what could happen in the future, I do believe that neurobionics, like Paris and London in the novel by Charles Dickens, it's really a tale of two cities. One city, as I said, is neuroscience, and the other one is the clinical application. So we tend to have neuroscience discovery and principles being transferred into neuroprosthetics, into devices to help the quality of disabled people. But at the same time, more and more, it is clear that it is also the other way around. So when you have a device implanted into the nervous system, then you can understand more how we work and how the nervous system works. I will just give you one example. For example, we have a collaboration with a great friend of mine, Andrea Moro, who is an expert in language. And what we are working with him and the hospital Niguarda in Milan is to use the stereo electrodes implanted in the cortex of patients for epilepsy to get signals during different kind of specific syntax stimuli, language stimuli that we can create. So if you want, the technology was used to help the disabled people, the people with epilepsy, to improve their quality of life, but at the same time can open a window into the nervous system that people like Andrea Moro can use to understand more, for example, language. More specifically, what we do in our lab, is a bunch of different things. I'm going to show you mainly what we do on limb amputation, but we've been working a lot on spinal cord injury. We are currently, for example, exploiting an exoskeleton in stroke survivors to see how these guys can benefit by the use of a personalized robot. Or, for example, here, completely different goal, we learn with Dario Floreano how to control a flying drone you're going to see in 1 second by moving the trunk. So the idea is to exploit our knowledge for neurorehabilitation, but also for other kind of applications like the flying drone with my friend Dario Floreano, the PFL. More specifically, I think you all know this video. Today, I'm going to show you what we did in the past 20 years to restore hand function in amputees, exactly like what happened to Luke Skywalker after somebody related to him cut his hand. I'm not supposed to tell who was the guy by contract with Disney, so you will discover later if you see the movie. In particular, what we've been working on is the possibility to create an interface with the peripheral nerve, which is kind of a highway connecting the brain, the central nervous system, with the periphery, in particular the muscles of the hand to control the movement, and at the same time, all the sensors which are embedded in a natural hand which are providing tactile position information, everything about the movement of the hand and the interaction with the external world. In particular, today I'm going to show you what we have been working on for the restoration of sensory feedback. The very first, let's say, step, and I'm going back to this later at the end of the presentation, was to find the killer application. So to find a way to interface the nervous system, the peripheral nervous system, with enough richness, so a good selectivity, being able to create a good relationship with the peripheral nerves, but at the same time with a reasonable invasiveness, because we don't want to be too invasive. This could prevent a real clinical application. So it took us 20 years to go up and down with different ideas. And finally, with Thomas Stiglitz and a student in his lab at that time, Tim Boretius, we came with the idea of these time electrodes. The idea is pretty simple. I don't know whether you can see it, but there is a needle, which is done in tungsten in general. You enter with the needle transversal into the nerve, and then you leave in the nerve a structure which could be polymeric structure, polyimide, something soft and flexible, which then has small dots, and these dots are the active site which can generate the current, which then can be used for delivering the sensory feedback. So it took us a lot of time, but when we found this solution, we decided to go for a first short-term 4-week clinical test with Paolo Maria Rossini, who is the neurologist I've been working with for more than 15 years, and with a bunch of people who were very young students at that time, and now they are more or less all of them colleagues of mine in different countries. And the idea is to implant, you can see here an example with the needle penetrating the nerve. Is a reasonably simple, it's not super simple, but it's a reasonably simple task. It took a few hours to the surgeon, even if you need to have need to be a bit careful in the implantation. You can see that for the surgeon, it's not super complicated. So we implanted 4 of these electrodes, 2 in the median and 2 in the ulnar nerve, which are the 2 main nerves connecting the sensors in the end with the brain. And as I'm used to say, there is the moment when you switch on everything and you try to see and ask the subject, do you feel something? And the very first time with Dennis, this first patient, it was kind of a miracle. You know, you switch on and you start stimulating the different dots, the different channels of the time electrodes, and then you ask, what do you feel? And Dennis was able to refer sensation at the phantom end. So he has a transradial amputation, an amputation at the level of the forearm, meaning that there is no more the end, there is an artificial end with sensors, but he was able to feel that these different stimuli, the current delivered through the electrode, were eliciting different tactile touch sensation in different parts of the fingers. So that was the first very interesting result to us. And then what we found is that increasing the charge, the amount of current we were delivering, we were able to increase the sensation. So he was able to feel something at a specific location, for example, on the little finger, but with an increased pressure generated by an invisible object. Okay. So then the question was, it is possible to exploit this in real time. So can we ask Dennis, the subject, to grasp some objects using muscular activity, so contracting the muscles, having the prosthesis closing around the object, with sensors in different fingers, then translating the information gathered from the artificial sensors into stimuli. In this case was amplitude modulation, meaning that the more you were getting sensation, the more from the artificial sensor, the more we were increasing the current in the electrodes. And then we asked Dennis to perform a huge amount of tasks. Just to give you an idea, it was more than 700 trials in a week. So we did more than 100 trials on average per day. So the first thing was to ask him to modulate in a staircase way up and down the force, you can see here, and also to look at whether he was able to produce different level of force, high, low, and medium. In this case, he did it statistically in a significant way, but if you want, was also interesting to see, you can see the red arrow on the right on the screen, that when he was realizing that there was kind of a mistake in the modulation, he was able to stop it and in real time readjust the force. So Dennis was able to use properly, at least in this task, this kind of information, but also in real time understanding what's happening and adjusting, getting countermeasure in case of mistakes. Then Dennis is a unilateral amputee, so he has still the natural hand, and we ask him to perform the same kind of staircase task in 3 conditions. With the natural hand, so relying on the visual and tactile natural information. With the robotic hand, without our tactile information, only looking at the object, and vice versa, only with our tactile feedback. And what you can see here is that more or less, we was able to achieve performance which were quite close to the natural one. There is a difference here, as you can see, between our blue results and the green ones, which was the natural one. The main reason for that, I'm going to tell you something at the end also about that, was the limited quality of the sensors we were embedding. So then we started playing with Dennis with a lot of different tasks. I will just give you few examples for the sake of time. We can talk about it later if you want. So we asked him to identify how stiff were the object, more or less stiff, and he did quite well in terms of performance, even only with 3 sessions, with kind of an improvement over time. And if this works, I want to show you, this is Dennis grasping. Of course, he was completely blind and acoustically isolated. And he's grasping the different object, and as you can probably hear, he's able to say the kind of stiffness of the object. So we played a lot with him on this, and also we asked him to recognize different shapes of different objects. A large cylinder, a small sphere, or a baseball ball. And again, he did it with very good performance. The interesting thing was that it was not his ability to understand what was happening was not due to the final level of force generated, but to the trend in the force over time. Something similar dynamically to what we do to exploit our natural tactile information. So we were able to say that he was able to use it in real time, and often in a way which resembles significantly the way we do in real life, okay? So then we started playing a bit more, in particular with Calogero Oddo, a colleague of mine at Scuola Sant'Anna in Italy. He developed a new kind of sensor which is able to get 3-dimensional information related to the touch, and we use it to understand something a bit more sophisticated, if you want, the pressure, but textural information. We started with 2 different gratings. So Dennis was asked to understand whether one half or the other one had a more dense grating. But then recently we published with also another colleague of mine, Alberto Mazzoni, another paper showing that 2 additional subjects were able to understand the kind of natural object. So not only gratings, which is a bit artificial, but understanding, not using, for example, temperature, but only, because we cannot restore it yet, but only touch, they were able to identify pretty well, one in particular extremely well, different kind of natural textures. So just to give you an idea of the amount of things we can do and we could do in the future using this technology. Then, of course, the next step for us was, okay, we did a 4-week implant. Can we do a long-term implant with the patients? Before doing that, Sophie Wurt, a former student in my lab, performed a huge long-term chronic validation with rodents. And in a nutshell, if you want, the results were really good, meaning that you get long-term usability, long-term stability. You can see here that after a few weeks, as expected, the electrodes are electrically very much the same, both in terms of threshold to deliver, a sensation, and in terms of impedance. But also the fibrotic tissue, which is unavoidable, tend to reduce the level of inflammation after a bit of months. So let's say we were reassured that it was reasonable and ethically possible to do human implants for more than 1 month. And in fact, we implanted 3 patients between the end of 2015 and the end of 2017. Here you can see 2 of them, Eduardo from Ecuador and Almerina from Italy. And the very first thing that we did was to replicate all the results that we achieved with Dennis. So to be sure that we didn't get the lucky card, but that what we were doing was really reliable for more than 1 patient. More than this, or more interesting clinically than this, was that we have chosen 3 patients with very different performance, very different history. For example, Almerina, this old lady, she got the amputation almost 30 years before the implantation. Eduardo had a huge phantom limb pain, really terrible, unfortunately. So, but the take-home message was that with all the subjects, notwithstanding the different performance, the different characteristics neurologically, we got very good performance. Even more interesting was the fact that with 2 subjects, Almerina and Loretana, the last one, when we started the magic moment of the starting of the stimulation, most of the sensations were related to the stump, not to the end, which was very bad for us. Over time, both of them, in a way or another, recovered most of the tactile sensation at the end. You can see here, both for Almerina and Loretana. So the message is, it seems there are only 4 subjects. Of course, there are not 50, but it is reasonable to say that this is a very reliable approach we can exploit later. And just for curiosity, 2 examples. This is Marco Controzzi, a colleague of mine with one of our subjects, performing what we call a virtual egg test. So the idea is to have a fragile object which has to go A to B and without breaking it. Of course, the advantage is that you cannot break it also or thanks to our sensory feedback. So we put a lot of the task. Here is not perfect, but there was an improvement in the performance. And in general, when you see this kind of test, you see always kind of a zoom on the subject, but none related to the rack outside. And there is always a lot of electronics, which is not really implantable, wearable, or portable. So what we did as a step toward the fully implanted system was to have all the electronics for all the 3 subjects embedded into a backpack, and the 3 guys were going when they went outside of the lab, outside of the hospital, going around Rome, enjoying all the. For example, Fernando, you can see here, was driving. You can see also the face of the neurologist behind was a bit concerned, but the end was they were there, they're still friends, and the car was intact. So just to say that the technology is already reasonably robust for this. Of course, the next step we are working on together with Soleiman also is to have our first fully implanted device implanted in Dennis, the first patient I told you about, in more or less 1 year from now, if the COVID situation is going to improve. So this will be a very important clinical goal, of course, but in parallel to this in the next 10 minutes, I want to show you two things. One, the idea that, again, as I told you about the tale of the two cities, we can use this opportunity to understand more what we can do in terms of science. For example, embodiment, which is the ability of the subject to feel this new artificial part, or as embedded, included, incorporated in the natural schemes, or other things that I'm going to show you in 1 second about increasing the amount of sensation we can deliver. In particular, with Edoardo Danna, who is now a postdoc in US, and a colleague, Alberto Mazzoni, we try to do something that most of my colleagues consider very bad, even to think about. So in general, the idea is you stimulate what you get, so what the patient is referring, you exploit it. If there is something the patient, for any reason, is not able to feel, that's it. You are going to lose something. Since position information, proprioception information are very difficult to be generated by natural stimulation with the peripheral nerves, we decided to have a mixed approach, using for the touch information what you just saw before, so the natural evoked natural sensation, but then using other channels which were not used by the patient, and teaching the subject a remapping of the sensation. So the subject was maybe getting some tingling at the wrist, and we explain her, the two ladies, them, how to translate this tingling into position information. Okay, so it was kind of a remapping. Then we asked the two ladies, first, can you discriminate the size of the object according to this remapped position information? And they did it, as you can see, reasonably well. Then a colleague of mine, a friend of mine, kind of challenged us, said, okay, but you will not be able to use together tactile and position information because this is going to mix everything up. And it was wrong. We asked the subjects to understand the softness and the size of the object, and they did reasonably well. Remember, this is becoming a task which is more and more complicated, even for healthy hands. But they were able to do it. And then, of course, the same guy challenged me again, and he told me, if you ask the subject to do this, they do it because they are very focused. If you ask them to do something else, they are going to fail. So what we did was to ask the subject to do a dual task. So they were doing the task of squeezing balls and understanding the size and the softness, while doing memory test cognitive task. So the idea was to simulate as kind of a divided attention, something more ecological, something which could happen in the real life. And here you can see the results are really interesting, because if you look at the size identification, you don't get any change between C, which is cognitive, and no label, which is no different, so no cognitive task. The only difference, which is not statistically different, significantly different, but it is big, it's not on the position information, but on the tactile stiffness task. Why? Because getting the stiffness of an object could be complicated. So if you distract the people, there can be mistakes. But the remapped sensation was still available as before. Okay? And then we decided to go a bit more complicated. I'm going to the end very quickly. A bit more complicated in what we were doing, asking a colleague of mine, Sliban Besmaya, to team up together and use his biomimetic model. So he developed a model he published in PNAS a few years ago that can translate any stimuli to the simulated fingertip into a biomimetic response of the nervous system. So it is a model of the nervous system. We generate an output in terms of change of frequency, which resemble the natural one. So what we did was to use this information, so the frequency modulation bio-inspired, or the amplitude modulation, or a combination of both. So to see whether you could get better performance in the virtual leg task and a more natural sensation, okay? And together with Giacomo, who is now a postdoc at ETH Zurich, and again Alberto. Here you can see Almerina, the subject. Hopefully you see Almerina, maybe. Okay, maybe you're going to see. So the take-home message is you get more natural performance, better performance, more rich sensation if you combine the frequency modulation done by the model of Schliemann-based Maya with our amplitude modulation, okay? This is just, again, a hint of what you can do making something more complex and more sophisticated. It is clear to me that we are just scratching the surface, and we can do way more than this in the next future. At the same time, the cool thing is that now we know that we have a technology, this time intraneural electrodes, which could be extremely powerful for different applications. So you saw hand prosthesis. We also implanted them with a former student, now colleague of mine, Stanislav Popovic, in 3 patients for legs, for lower limbs, getting even better results. But now I launched this, what I called Eye for Life project, which is doing more than this. So going from amputees, both upper limb and lower limb, to other applications like visual prosthesis or restoring sight in blind people, restoring grasping movement in paralyzed people, and electroceutical, which is modulating the autonomic nerve. I will tell you something in 1 second. So first, optic nerve stimulation. A few months ago, we published a paper with my colleague Diego Ghezzi at EPFL, showing in rabbits that we can elicit very rich and independent sensation in the cortex of the rabbit using different active site of the time electrodes implanted in the optic nerve, meaning that it seems, of course, with rabbits you have a bit of limits of what you can do. It seems that you can restore a quite rich visual sensation in rabbits using the time electrodes. And we are starting, we got a small grant from an Italian private foundation. We are going to start in Rome again implants in blind people of the same technology. You know, the advantage is again that what you get, what will be done to have a solid and robust solution for amputees now can be exploited for the optic nerve. It can be exploited also for grasping restoration in paralyzed people. So instead of stimulating the sensory nerves in the upper arm, you can stimulate the motor nerves after a paralysis and restore grasping. We started with excellent results in human primates, and now we are ready to have probably next year the first patient with spinal cord injury, with a cervical spinal cord injury implanted. Finally, electroceutical is a new idea, a new concept of interfacing not the sensory motor nerves, but the network of the autonomic nerves, which are connecting the nervous system with all the internal organs. In particular, just a very last comment, we got a European project I'm coordinating called New Heart. And the idea of New Heart is to restore the connection between a transplanted heart, when it's reimplanted in the new subject, and the vagus nerve. Because the vagus autonomic nerve is kind of the brake of the car. So it's down-regulating the cardiac activity. All these subjects, because they don't have any kind of vagus control, they tend to overuse the heart, and this is going to damage after a few years the heart, getting them into the need for a new transplantation often. So again, the message I wanted to convey is first, intraneural stimulation is a very interesting solution for the restoration of sensory function in amputees, but also what we are going to do now is to exploit the same kind of technology also for other applications, for blind people, for paralyzed people, and even for all the electroceutical vagus nerve stimulations. And with this, I think I can hopefully stop sharing and come back live. I hope that you hear me. Hello?
Hello. So thank you very much, Dr. Misera. It was an amazing presentation. And so since we have a tight schedule, let's jump directly to Solomon's presentation, and then we follow to questions, okay? So let me just introduce Dr. Solomon. Well, Dr. Solomon is currently a senior scientist at the Bertarelli Foundation Chair in Translational Neuroengineering in Geneva. Previously, he worked with the development of virtual reality-based brain-machine interface for hazardous monkeys and closed-loop brain-machine interfaces that was integrated both motor and tactile functions. From 2015 to 2019, he was the research coordinator at Alberto Santos Dumont Association for Research Support in São Paulo, Brazil, where he was in charge of the development of neurorehabilitation tools for patients with spinal cord injury. In 2019, his work was nominated for Brain-Computer Interface Award. Dr. Shokur has obtained his PhD with distinctions at the PFL in 2013 under the supervision of Professor Miguel Nicolelis and Hannes Bleur on the development of the first virtual reality training system for non-human primates. From 2010 to 2012, he was a visiting scientist at Duke University. Well, also in 2014, he was chosen to represent the Walk Again project for the opening ceremony in the World Cup, where a spinal cord injury patient trained by Dr. Shokur and the Walk Again project team used a lower limb exoskeleton for a symbolic first shoot. So it is with great pleasure that we have his presentation titled Recovering Somatosensory and Motor Functions with Brain-Machine Interface, Advances in Assistive and Restorative Technologies. So Dr. Solomon, thank you very much once again, and the word is with you when you want.
Very much. Can you hear me? Can you hear me? Hello? Hello, hello?
Yeah, it's working.
Yes, and can you hear me actually?
Yes.
No? Yes, you can hear me? Okay. Well, thank you very much for the presentation. Thank you very much for the invitation. Great pleasure to be here. Wait, let me see if I can still. Okay, so my presentation will be a bit the continuation of what Silvestro was presenting. It's sort of hard to continue after Silvestro's presentation, I guess. It's a bit humbling after all what amazing stuff that he has been showing. So the idea would be to continue on the idea of recovering somatosensory and motor functions, but looking more from the brain, targeting more the brain. So using brain-machine interfaces for recovering motor functions, and also new techniques that integrates brain-machine interfaces with sensory feedback. And well, also continuing with the same analogy of the Dickens book, I will also be showing both the assistive part of this technology and the restorative part. So how these techniques could be used to assist patients to recover this function, and also how throughout training, they could eventually recover some neurological functions in their body. So we'll be talking about spinal cord injury patients. I always like to start with this slide, remembering that spinal cord injury patient, the typical patient is a young male adult. The average age is 32-year-old. Thanks to advances in surgery and medicine, the mortality of a spinal cord injury patient, actually the morbidity is low today, so this patient will survive to the lesion, but then they will survive, they will live for 20, 30, 50 years of their life in a very, with a very hard deficiency, which will limit their quality of life and their activity of daily living. It's also important to remember that a majority of these patients are considered or diagnosed as complete lesions. So that means that this patient will lose all sensory and motor functions below their injury. So these patients, this is important to actually distinguish that these patients are clinically complete. So what we call a complete patient is this patient who have no clinical signs of sensory and motor functions below the lesion. That doesn't necessarily mean that they have a clear cut at the lesion. I just make this distinction because it will be important further in the presentation. Globally, there is about 25 new cases per million per year of spinal cord injury. I did a bit of my homework. I looked this morning in Brazil. The cases are slightly lower than the average at the world level is 70 new cases per year per million people. Now, in the last about 20 years, brain-machine interfaces have been proposed as sort of a radical solution to restore motor function in these patients. So as Silvestro showed very nicely, for amputee patients, there is a lot of interesting research and actually very mature technology to record muscle or nerve levels to control, say, a robotic arm. Now, for a spinal cord injury patient, unfortunately, we don't have that opportunity. So the idea has been to record really at the source, so record at the brain level, decode motor functions, hopefully in real time, or let's say quick enough, to control what's happening below the lesion, or either a robotic device or exoskeleton, or to stimulate the muscles. And now, one of the most important results that are still used today for, or discoveries that is still meaningful for brain-machine interfaces development today, was by Georgopoulos and Schwartz in the mid-80s. Georgopoulos and Schwartz actually were recording neurons in the motor cortex and found that the encoding of the motor functions, so the encoding of the position of the end effector, in this case of the hand, is surprisingly simple and straightforward in the motor cortex. So what they did is they trained a monkey to play with a little joystick, and the monkey was moving this joystick around, and they were recording neurons in the motor cortex. What they found is, this is an example of a certain neuron. These are little spike trains that I'm sure you're familiar with. So this is 5 trials when the monkey was moving this direction, and then several trials in different directions. In this specific neuron, for this specific neuron, here is the 0 time. The activity of the neuron increased when the monkey was moving in this direction. So it would be 135 degrees. And then for different directions, this increase in activation was less important, and in some cases even was lower. And looking at the different movement on different directions, this follows a beautiful bell shape here, a cosine, and with a peak around 135 degrees. So what they call this is that this neuron has a preferred direction in this specific direction here. If you look actually carefully, the activation here actually even happens a few milliseconds, about 200 milliseconds before the monkey starts moving, which means that if you record this neuron, you will know 200 milliseconds before the monkey moves that he will be moving. You can predict that in advance. The next few years, following pioneer work by Nicolelis, so I think a well-known Brazilian neuroscientist, who developed techniques to record neurons in various neurons at the same time in various different areas of the brain using multi-unit electrodes. Using this technology, Carmena and Nicolelis and colleagues did the first demonstration of a BMI using implants. In this case, the monkey was also trained to use a joystick, but instead of recording just one neuron, they recorded, in this case, if I'm not wrong, 40 neurons in M1. What they found they were recording was the kinematics and the neurons, is that maybe neuron 1 had a certain preferred direction to the left, neuron 2 had a different preferred direction, etc., etc. And all these neurons, they had different preferred directions. Using all of them together, they could build a prediction, just using a simple linear model of this firing rates. They could build a prediction of the 2D position of the hand just by looking at the neural firing rates. Using this approach, 2 years later, Velis showed this demonstration here. You have a monkey here. I don't know if you can see, you can perceive the video. So here the head is a bit hidden because he has an implant in the brain. And the monkey is brain controlling this robotic arm in 3D in position and controlling also the grasping for self-feeding. So to resume, what we know is that there is a consistent correlation between neural firing rate and specific behavior and activity, and specifically in M1. The M1 neurons respond with peak of activation in specific directions, which gives this neural population vector in 3D. And using a neural population, at least 10 to 30, but later on other researchers have been showing that even you can use 100 or 200 neurons, you can have even better prediction. Using this is possible to have an adequate brain-machine interface control. Now here's another demonstration with a human subject, a clinical test, using exactly the same sort of approach. Here, this is a tetraplegic patient with an implant, a new array implanted in the hand, in the motor cortex, who's controlling again the 3D position of this robotic hand and also the grasping. While this research is the result was quite spectacular, what we can notice by looking at this video is that something is missing. This patient has a lot of difficulty actually to perform this task. I don't want to diminish for sure this result, which is outstanding as the patient can control this hand, but you can see that the patient has to rely a lot on vision, has constantly to look at it. And if you look actually carefully in this, unfortunately my video doesn't permit me a smooth, nice image. The patient has really hard time to grasp and to know when he has a good grasp of the object. So I guess it will not come as a surprise to you that I believe, as Sylvester has also showed with this amputee patient, that tactile feedback, me and Nicolelis and other people working in this domain, that tactile feedback is essential also for brain-machine interfaces to move to the next step. For the patient to have a better control, we need also to give sensory feedback for the patients. Now, why is it important to target both somatosensory and motor recovery? Well, first, as we have seen in the previous presentation, this is essential. Tactile feedback is essential to experience the world, to recognize shapes, texture, object softness, etc. Remembering also that the vast majority of patients who have lost motor function in spinal cord injury patients, they also have diminished or loss of sensory functions. So it makes sense to give them both the motor and sensory functions back. Second, because it's important for motor planning. Having the tactile feedback is essential for the patients to perform dexterous movement. Third, because tactile feedback is important to reconstruct or to construct, and update the body representation in the brain. So the brain representation of the limits of the body and the immediate surrounding of the body, called the peripersonal space, needs the congruent information between vision, tactile information, and proprioception. So it is important also to give this feedback for the patient to have a sense of embodiment of the prosthetic hand. And finally, because under some circumstances, and I will show you some example of this at the end of my presentation, we have observed that this could induce a protocol using both sensory and motor functions could induce neurological recovery. Okay, so how we can give the sensory feedback targeting the brain. Here, one of the most important research were done by, or one of the pioneer work was done by Romo and colleagues in the late 90s and early 2000s. What they observed is that, so there's a sensation of fluttering that you can generate in a monkey. So placing a little vibrator on the monkey's hand, you can, and by simple mechanical vibration, you can create a sensation of fluttering. And the monkey was trained behaviorally to receive two stimulation, stimulation one after the other one, and he had to respond which one of the two had a higher frequency. While they recording neurons in S1, they observed that actually there are neurons in the somatosensory, primary somatosensory area that respond to this fluttering stimulation in a somatotopically, say, similar way to the stimulation site and to the stimulation frequency. Say differently, if you record in S1, you could actually predict or detect what is this, which part of the hand of the monkey was stimulated and with which frequency. Now, playing the game backward, they stimulated neurons in area 3B with various frequencies. And what they observe is that, at least behaviorally, the monkey responded similarly to the mechanical stimulation on his finger, and the stimulation, the electrical stimulation in these neurons in area 3B. What they conclude is actually it's possible to create different sort of fluttering sensation by manipulating the frequencies of stimulation in S1. Now, taking together what we have just seen with Carmena's, Nicolelli's work and Romo's work, in 2011, also in Nicolelli's lab, in a work in which I had the chance to participate, we tried to put both ideas together. So here we had a monkey playing with a little joystick here to control like an avatar arm on a screen. We recorded neurons in M1 to control the position of the avatar. The avatar was touching different virtual objects. And this object generated a virtual texture, which then stimulated back in the sensory area using intracortical microstimulation. So the recording here was done also with electrode implanted, M1 for the recording and S1 for the stimulation. On the next video, I will show you an example of tasks done by the monkey. You will hear a little BB. So you will see, this is brain control. So the monkey is controlling this with the brain. He sees this different virtual object. This is the correct one. This label didn't exist on the video, obviously. This is just for me to show you. Some people on YouTube said, okay, this is very easy. The monkey learned to read correct targets. And that was not the case. So this didn't appear. And you will hear sometimes a little beep, beep, beep. And that is the artifact from the stimulation. Okay, it looks like the video is very badly lagging. So, well, what you might have seen is that the monkey is exploring the different objects, feeling the sensation for them, and then when he finds the correct one, he stays on it for about half a second and gets his reward. Now, this is actually quite tricky to do. You can hear it also through those beep beep, which are the artifact. What happens is that while we stimulate in S1, because S1 and M1 are quite close, we are completely saturating the recording in M1. So if you're trying to decode something from M1, what you actually see is pretty much nothing. You simply have saturated the signal there. So the trick we played here was to intermingle the periods of stimulation and recording. So we were multiplexing. Every 50 milliseconds we will stimulate, we will give the title stimulation, and every 50 milliseconds we will record from M1 to build our decoder. Last year, we went one step further with this research by creating the same idea, but the same idea of a more advanced version of this texture feedback, but using an active tactile exploration. So if you think actually on our 2011 study, the stimulation was some sort of a buzzer. So when the monkey was on top of the object, no matter the position of the avatar hand, the stimulation was always the same. So it was almost like a buzzer that will stimulate at a continuous frequency. Well, this is not how we experience texture in our, that's not how we experience texture in our daily living. So if you move your hand, let's say on a carpet, on an object that has a texture, your sensation will depend of the speed of your hand. So if you stop moving, you probably won't feel much. So that's what we try to reproduce in this experiment. So every time the monkey has, at each moment, the monkey has two objects. One of them has a higher spatial frequency, like regions that are closer, and one of them has lower spatial frequency, and the monkey has to explore both of them and choose the one that has the highest frequency. I don't know if the video could play. This is... Okay, I'm gonna give up on the videos because it's painfully slow, but okay, believe me, the monkey is managing to find the one that has the highest frequency. We tried many combinations of regions. So many combinations of left and right object regions. What we found obviously is that if the frequency between the two objects was very different, the monkey had no difficulty to discriminate them. But when the two frequencies were getting closer, this is cases where the monkey started to confuse a bit more. And interestingly, when the frequencies were higher, the monkeys had even more difficulty to do it. Simply said, a monkey could discriminate easier between 0 and 0.5 in frequency than between 3.5 and 4. So the just noticeable difference was a function of the standard frequency, which follows Weber's law on perception of natural sensation. Another interesting observation we had here is, so because the stimulation pattern was a function of the speed, we had what we call paradoxical trials. So imagine a case in which the monkey has to move over these two objects. This one has 2.5 regions by centimeter. This one has 3.5. But let's imagine that by chance he moves very quickly on this one and slowly on this, which is something that happened in a subset of the trials. Well, if we look here at the profile of the speed and the position here, if you look at the profile of the stimulation, in this case, the monkey received a higher frequency stimulation on this one compared to this one. So here we have a paradoxical case in which he actually had a higher stimulation here, but the correct one was this. Well, what we found is that in at least one of the two monkeys, this monkey was able to correctly choose the right one, which was this one, significantly above chance level. And this is a case where the intracortical microstimulation alone is not enough for decoding of texture, but the monkey had to integrate the intracortical microstimulation plus the avatar's speed in order to decode or to understand what this decoding was. And this indeed worked, worked significantly above chance for one of the two monkeys. Okay. Next work, this is work by a team in Pittsburgh, Ganser and colleagues. This is a paper that came out a few months ago, which I think is quite interesting. Also, to give you a bit of a flavor of what's going on in this domain, we have been working at Nicolelli's lab with monkey models, but since a few years there are clinical tests also with patients, with tetraplegic patients. In this case, the researchers implanted electrodes also in M1 and used a functional electrical stimulation to move the patient's hand, and recorded and decoded, actually, the tactile functions, or the tactile the detection of tactile the detection of contact of the hand with objects through the same BMI. So the patient was an Asia A patient with a C5 lesion. He had a full range of motion up to the elbow, but no ability to grasp. What they found is that recording in M1, so this patient has no sensation in his hand. But the interesting part here is that the team found that it's possible to record some signals in M1 when the patient is touching certain objects. So although the patient cannot feel it, that signal exists and it's possible to detect it. So reading in M1, they could actually detect when an object was touching the forearm, the thumb, the index, or the middle finger. Now they use this to, I think we can see here a little video, but again, videos doesn't work very well. Now they use this decoding to amplify the sensation on the upper arm. So remembering that this patient had sensation in the upper arm. So this, when the patient was touching an object, this was vibrators were vibrating on or were stimulating the patient's upper arm. The interesting part with this is that they found that so having the patient doing the BMI plus tactile feedback significantly improved the function of the patient. So the patient was better at reaching object, grasping object, and maintaining a stable grasp. So I think they used here the ARA test, and you could see that the object transfer time when the tactile feedback was present was significantly lower than in absence. Now, on a different, with a different approach, also in our team in Brazil, so this was done in an experiment done in São Paulo. We used brain-machine interface for paraplegic patients with functional electrical stimulation to reproduce locomotion. So here, the patient imagines moving his or her own leg. We detect motor imagery from here, and we use it to stimulate the lower limb to produce for producing a gait. I'm going to skip a little bit on the technical part of this and show you a video. So here, this is a patient who was originally an Asia A, so a patient who had lost complete sensory motor functions. This is after 4 years after his lesion, so he is at the chronic phase of the lesion. So here we use this EEG system to decode motor imagery and to control the stepping of the corresponding leg. On the BMI side, we used a non-invasive technique, so 16-channel EEG system. The patient first trained in a virtual reality system, so where they were trained to control the stepping of an avatar, and next they moved to the VR. Now, for here we have an example. I think I'm gonna again, fortunately, have to skip these videos, which are not very visible. Now, on the sensory domain, we used arrays of vibrators aligned on the patient's forearm, remembering that these patients are paraplegic, so they had full functions on the forearm. And these vibrators were informing the patient of the position of their leg in space, so they had a continuous sort of sensation on their forearm while their leg was swinging in the air, and then sort of a shock on their forearm when the foot was in contact with the floor. Now, using this approach, training the patient for several months, we observe this sort of effect. So this is a patient with T11 lesion, so thoracic 11 lesion, 12 years post-lesion, so also at chronic phase. And this is after 13 months of training. This patient has recovered sensory function of the toes, so could actually feel sensation in the toes, and also could contract some of her lower limb here to do an adduction, against gravity. Here, another example for a patient performing a flexion, also against gravity. The cables that you see here are not stimulators, but in this case are just EMGs to make sure that the patient is not using some sort of compensatory mechanism. And here was another example also. So this patient was an Asia A at the beginning with a T11 who could perform significant contractions. We observed that in almost all our patients, so consistent electromyography responses, muscle responses in the lower limb, recovery in the sensory domain, and this recovery was sometimes several myotomes below the anatomical lesion. So we checked with MRI the anatomical lesion. This recovery wasn't just around the lesion, but it was sometimes up to 10 myotomes below that. As a result of a long-term protocol, this was a 28 months long protocol with 6 patients, with Asia A and 1 patient with Asia B. We observed an improvement. So Asia A, is patient that have lost complete sensory motor functions. Asia B is a patient that has sensory but no motor, and Asia C is a patient that has recovered sensory function and some motor functions below the lesion. So what we saw is that a systematic improvement of all the patients. The only one that didn't improve is a patient that had stopped the training, dropout from the protocol after 12 months. So this is, to our knowledge, the first time that a study was showing a systematic improvement in patients with most severe cases of lesion of Asia A. Jean, tell me, am I actually late? Can I have maybe 2-3 more slides? I didn't really follow the time.
Yeah. You have
5 minutes?
3 minutes. Yeah, it's okay.
3 minutes, okay. Okay, so
5 minutes as well.
Just 5 minutes. Okay, perfect. So I think there's plenty of time. So one very interesting result, at least for me, that it's very exciting for me, it's concerning the sensory representation. We saw that also a little bit with Silvestro's work using the implant at the nerve level. We observed changes in patients' representation of their own body. Here in this case, this was a patient with a lesion at T4. And the test was done with vibrators placed on different parts of the lower limb. And the typical clinical question is, the clinical task is for the patient to tell you if he could feel or not the stimulation. And this patient, at the beginning, had no sensation at all, and after 4 months, 7 months, and 10 months, started recovering sensation to vibration. So this is the first part of the test, of the test. What we did is that we tried to go one step further and actually ask the patient, where do you think the stimulation was done? So we would stimulate in these different parts, and we will say, when the patient is with eyes closed, we say, where do you think we stimulated you? So what you see here is the ribs, hip, knee, ankle, and foot. So you have the little circles here saying where we stimulated, and if there's a line, it tells you where the patient has felt the stimulation. So at 4 months, the patient was feeling nothing. After 7 months, the patient started feeling the ribs, started feeling the hip, but when we stimulated the hip, the patient had the impression that he was being stimulated at the knee level. And this was happening both on the right and the left side. And this effect actually continued over and over. And after 16 months, here the patient, when we stimulate at the hip, he feels the knee. When we stimulate at the ankle, he feels the feet. And this is quite striking, actually. And the hypothesis we have is that once the brain starts getting this new information from the lower limb that didn't receive since after the lesion, this body representation is all messed up. So the patient starts feeling the hip and he says, okay, this must be the end of my body. This must be maybe my knee. Or when he starts feeling the ankle, he says, this is probably as far as my body can go. So he feels that it is the foot. Interestingly, if you continue the training, at some point, this sensory map starts getting more reliable. Okay, so this is my last slide also from just presenting you what is happening outside our lab in Brazil. So we did a lot of work with Nicolelis and colleagues in Brazil with non-invasive techniques. There is a team of young researchers, I would say young professors at the University of Pitt, who are very active using implants to control robotic arms and implants for giving sensory feedback. They have some results. I mean, the main one the most interesting one that I have here is not yet published, where they also find that by using tricortical microstimulation with human subjects, there is an improvement in function. So the patient is faster to grasp, and he has a better sort of dexterity. The absolute last slide, just to also give you a little bit of idea of where the domain is going. You might have heard of Neuralink and the new developments of the new startup by Elon Musk, who is aiming to make an electrode implanted in the brain. Well, it looks like, from at least what we know, the little that we know, is that they are also going for implants that can both read and write in the brain, and they will be targeting both sensory and motor functions. So it looks at also a Neuralink. They are seeing this future in which brain-machine interfaces will have both read and write capability in both sensory, motor and sensory functions. And with this, I hope I haven't been too long. Thank you very much for your attention, and I'm done.
So thank you very much, Solomon. It's also amazing as expected. I'm pretty sure that everyone has enjoyed it a lot. So let's jump directly to the questions. We already have some, actually a lot of them. We'll try to make one question for each one. So Daniel has separated some of them here. Well, so actually the first one is just a comment. Awesome, for both of you actually, awesome project that's going to help tons of people around the world. Thanks the effort, Professor Messera and Solomon. Anderson Villar, I was wondering if this treatment is just for hands and fingers, or if it is also for any part of the body. I think it's for Professor Messera.
I mean, the approach is usable for any kind of amputation. I mean, we already have evidence for transfemoral amputees, so for leg amputation above the knee, and it can be used for, the main issue is that, of course, the more you have a high-level amputation, the more difficult will be to find the right nerve to interface. So if you go for shoulder amputation, you can do something, but it becomes more complicated, and maybe other solutions like target muscle innervation could be a good solution. But if you want, the beauty of what we have now is that these implants, these time electrode, can be used really for a variety of different applications.
Yeah, perfect. So for Solomon, can the sensory motor cortex be covered with peripheral electrical stimulation? Also, do you believe we could use the same results to rehabilitate memory in frontal cortex?
Okay, so can we use peripherally, say using what Silvestro has been showing, for example? Is that the first question, I guess?
Yes.
Yes. I mean, it's not very practical, let's say. I mean, if we are talking about spinal cord injury patients, they generally have lost sensory and motor function, so it's simply not possible. I guess you can imagine doing it above the lesion. So if a patient has a C5, then you implant in the shoulder. I don't know. I mean, this is actually an interesting maybe idea. I don't know. It doesn't seem to be very practical, but could be an idea to investigate. Now, for stimulating in different areas, there are some groups are stimulating in different areas. There are people that are stimulating in the posterior parietal cortex, and they showed that it was working. I don't know about, I mean, I know that for sure for that, I know that people have been stimulating in M1 also, and that also worked. I don't know about other areas. I don't know if Silvestro has seen or heard. I mean, what is interesting with S1 is that there is no learning. It's a natural sensation, right? So it is, as Romo showed, you stimulate the same way that S1 responds to a mechanical stimulation of your hand. So it is as natural as it could be, I guess.
Yeah, perfect. So to Dr. Missera, Fabiola Alves would like to know if there is any accommodation for the sensory stimulus over time.
There is, yeah, I mean, there is the common problem, if you want, that you get accommodation. The interesting thing is that the accommodation seems less important happening later for amplitude modulation than for frequency modulation. And but in principle, it is very much subject dependent as well. So it is there, but it's not precluding the possibility to use this kind of information for different tasks. And it could be possible to play a bit with the sensation we are delivering to cheat the brain in some way to reduce this issue. Not to eliminate it probably, but to reduce it, yes. Or the other option, for example, very simple, is to use different channels conveying similar information. So you can stimulate two fibers, one related to this position in the finger and one to this position. For the subject could be pretty much the same, and you can avoid this kind of issue. But of course, it's there.
And what about the damage on the nerves? Is there some damage long time?
No, when we started working with intraneural electrodes, there were colleagues of mine saying, you are a killer, you want to destroy the nervous system, you are a bad guy. So maybe I am a bad guy, but not for this, okay? So in general, you get a damage, clearly, and it's unavoidable, but the damage is not painful. We have already now 8 subjects, including upper extremity and lower extremity, up to 6 months. None of them referred to any significant problem. So I think it is okay. I mean, I would say the question is, though, and that's something I have a bias, but we don't know yet exactly, whether the kind of sensation we can deliver with intraneural electrodes is better, more rich, and more usable than what you can get with electrodes around the nerve, a bit less invasive. There is a bit of evidence. I mean, if there were people like my colleague and friend Dustin Tyler, we could fight forever. But let's say that's the main issue, at the very end. Whether we get an advantage to be a bit more invasive, that's honestly a reasonable question. But you don't get really a serious damage to the nerve.
Yeah, exactly.
I could even tell you, sorry, that the stimulation can also reduce the pain in the subject. So most of the subject, like Fernando, the guy from Ecuador, he was getting a lot of pain, phantom limb pain, or Dennis as well. And thanks to the stimulation, he was getting a significant reduction in the pain. So this is a plus more than a minus, if you want.
Yeah, sure. To Solomon now. Well, Julio Roncato wants to know if the person cannot see the object, the signal will be made for the brain. I don't know if I got the question.
Could that be for the Gansel paper, I guess?
Yeah, maybe. Yeah, if it's not seeing, yeah, it's blind.
Yeah, so yeah, it's a good question. Like, have they? I believe that they. I mean, I'm certain they controlled for vision. The response they were having was only to tactile. So yeah, so the patient was blindfoldly touching objects, and the response they were looking in M1 was for tactile. And it's a good question because actually, if you had vision, I mean, there are some evidences also that you could have S1, for example, responding visual touch, I mean, under some conditions. And we showed that a few years ago with monkeys, where when we were touching the avatar hand, just with a virtual brush touching the avatar hand, we were having responses in S1. So there could be also responses to visual feedback. So it's an important question.
Yeah. So to Mesera, let me select one from my student here, Karina Rodrigues. She is my PhD student. So she wants to know what is the time required for training with this type of bidirectional prosthesis for the remapping of sensory information, sensory recognition, and to perform functional activities.
In general, it depends subject by subject, but I would say that on average is very fast. So just to give you an example, Dennis, the first patient, the short-term implant, I'm used to say that he is a plug-and-play person. So he was immediately able to master it. It was very fast, very fast. In other cases, it was more complicated, often not because of the subject, but because of some conditions of the subject. Just to give you an example, Fernando, I was saying he had a very, very high phantom limb pain. And then it was very difficult for us to stimulate above a certain threshold without getting pain. That's something you don't want to do, right? So we had a very small dynamics. And of course, the smaller is the range of current you can deliver, the more complicated it is to modulate it for the subject, both for understanding and for the control of the MG. So it took him a bit of time to learn it. But I would say, in general, it is extremely fast. Even the sensory remapping for the position, the two ladies or the texture, they were extremely fast in getting it, extremely fast. Because, at the very end, it's not perfectly natural, but it's very similar to something natural. So the subjects are pretty quick in getting how to exploit it. It is very, very easy, very fast.
Yeah. Yeah, I know why she's asking that, because we are seeing similar results with our project as well, but we are stimulating non-invasively. But we also saw a very fast adaptation in controlling virtual avatar. So
the trick will be then. So the trick will be then to understand how far they can go, because most of the tasks that we do are reasonably simple. So, I mean, pick and place and these kind of things. So if we ask them to modulate more, are they going to be able to do that? That's the main goal. But for something quick, they are reasonably fast in getting it.
Perfect, thank you. So to Solomon now, Guilherme Fernandes wants to know how is the brain signal decoded into movement, into mechanical, of the mechanical arm, I think. Our neural network uses lots and lots of signal cascades that ultimately lead to movement. So how is that created? Well, for you. Solomon, can you hear me?
Yes, I lost you after neural network. So I'm guessing this is about the sort of algorithm that are used. You can hear me?
Yeah, I can hear you. Let me repeat that. So essentially, he says, our neural network uses lots and lots of signaling cascades that ultimately lead to movement. So how is this signals or this coding recreated?
How is the... Am I understanding that this is about this sort of algorithms?
Yeah, maybe.
Or maybe if you get it in Portuguese.
Yeah, maybe, I don't know, I'm supposing. Maybe he wants to know a little about how brain-computer interfaces work.
Okay. So I think if we... So looking at those first few slides that I was showing, that is sort of the central idea, at least for invasive BMIs. So invasive BMIs, and this core always uses that idea that M1 is encoding the direction of movement of the hand, of the leg, etc., etc. Now, the transformation that you have there is between firing rates and a position or a speed of the end effector. And in the middle, what is the decoding process? It could be sometimes as simple as a linear regression, which is what was first shown in the paper by Nicolelis and Carmen and colleagues in 2003. And I mean, there is all the algorithms you can imagine. People are using artificial neural network, support vector machine, and then there is a bit of, I mean, there's a bit of families there, people who says that if you use Kalman filter, this is probably the best way you could decode. People will say, no, it has to be with a Wiener filter. But at the end, where we are at today is with these techniques to decode up to, I think the best ones can decode up to 8 degrees of freedom at 10 hertz, so 10 times per second, with quite high levels of accuracy. In one of our previous work, we did a bimanual task, so a monkey controlling 2 different arms at the same time. And this was using, again, a linear regression over the firing rate but we had a lot of neurons. That's one thing. In that experiment, we had 400 neurons. So I guess that also helped to have a easier, far better decoding.
Yeah.
Did I answer? Otherwise, I can send just a review for all the algorithms, because this is, or we can spend like a whole hour about it.
Yeah, that's it. It's not possible. So Daniel Leal, the guy who is supporting us, and wants to know how was measured the quality of the feeling that the users felt with this electrical stimulus. From your perspective, how long do you think this technology can be used more widely in clinical approach? I think it's for Silvestre Missera. Did you get the question? Okay, so I think it also works for you, Solomon. You can try to answer.
Yeah, so with the little drawback that in my case with monkeys is hard to know what they feel. So it's actually very tricky. But the jokes apart, actually, there has been some experiments where they were trying to stimulate mechanically and stimulate with electrodes. And what they found is that at least behaviorally, the monkey could not distinguish them, right? So the monkey had to respond when he was stimulated mechanically, and you could fool him or fool the monkey, when they were stimulating in the brain. So at least for those sort of stimulation, it looks that the sensation was similar. Now, I might be able to answer also for the amputee patients, as I'm working a little bit on that with Silvestre as well. Do you want to ask me what the question was for Silvestre? Maybe I can give it a try.
Yeah, sure.
So the question was how natural the sensation is. That's the question.
Yeah, how can you measure this quality of sensation? Because in some way it is subjective. No, so
absolutely, it is completely subjective.
Yes.
Yeah, it is completely subjective. There isn't yet a good technique that is more sophisticated than just the subjective question. So to the patients, does it feel natural? Does not feel natural? You can play a little bit by playing with different stimulation. You don't let know the patient, and you ask him how or which sort of sensation he or she has. But at the end, Silvestre, excuse me, I was answering to the question that was for you while you were away. On the subjective sensation of the stimulation, I guess at the end is always a question to the patient. How do you feel it? We don't have a better model than that.
Can you hear me?
Yes.
Yes, now, yes.
The connection is now. What I'm trying to say, yes, and in general, more or a question or at the end, the efficacy in terms of performance. In terms of nature, it's the only thing.
Silvestro, maybe if you reload the web page, the link can be restored in a better way. I don't know.
But if I can just add something while Silvestro is coming back, at the end, this is also a philosophical question, right? Of the sort of qualia, or what is your experience of red color compared to mine? Is it the same or not?
Yes.
It's a purely subjective question at the end, right? Of how you perceive a sensation, and I don't think there is a good neural model of that. I don't think. I mean, it would be ideal if we could record say in an fMRI and see when the patient is perfectly feeling a certain sensation, but that model simply doesn't exist.
Yeah,
not yet,
sure.
at least.
So let me make another question to Silvestro then to compensate this. I would like to know, this is from Priscilla Antonelli, students of mine as well. I would like to know from Dr. Missera, what is the biggest challenge in bionic limb development? It's a general question.
There are many. You can hear me, I guess, again. Huh?
Yes.
Can you hear me?
Yeah, I can.
I think a kind of one after the other. Now we elect are enough. But for example, the next is to have artificial more and similar to skin, and a prosthesis which can more degrees of freedom. So it's kind of cool. But we started they can provide with the now it's pretty good, and we are back to the robotics people asking them for more sensors, more degrees of control, and these kind of things. But now I would say the next bottleneck, the implantable stimulator and the sensor be embedded.
So, okay, I think the connection is not that good, but I think I got the general idea, no problem. So actually, we only have more around 3 minutes. I would like to open for you if you want to say something additionally from people that are watching us. Solomon, maybe.
Solomon, you want to start?
I'm sorry?
Yes, I think as you wish, Silvestro. I can go first?
Yeah.
So for me, just first, thank you for this invitation. Although it wasn't very easy with the videos and everything, I hope that we could convey the message. One thing I want to say also to people that are there is that we have some activities and some collaborations with Brazil. I'm personally a big fan of research in Brazil, and that's why I spent 5 years of my life in São Paulo. So if people are interested to also work with us, collaborate with us, you can just drop us an email. We have sent sometimes students there. We are receiving students from there here. And so we are more than happy to continue this sort of collaboration with you guys.
Yeah, very soon you're going to receive my students from here.
Yes, we're looking forward.
Yeah,
we look forward to it. I mean, just to echo what Solomon said, I don't know whether you can hear me, but it was really a great pleasure to join you today. I really enjoyed it. And I'm very happy to have the opportunity to show what we are doing also to you guys. I personally believe that neural engineering is super cool and will have an impact in the future. So if that you are thinking of starting working on biomedical neural, do that. It's a cool way to spend your period at university. And of course, we are very, very happy to start working together even more, exchange students. So thanks a lot. Obrigado, I guess.
Yeah, perfect. So guys, thank you very much. Wordless to say thank you for you both. It was amazing. I'm pretty sure that everyone has enjoyed a lot. Daniel also is saying here that was very, very nice for him, and he's also excited and expecting to meet you in Rio. So once again, thank you very much in name of Unifesp. Was amazing, and keep in touch, okay? So, tchau, obrigado. Thanks a lot.
Obrigado, bye bye.
Tchau, tchau. Tchau. Muito obrigado.